So today I want to talk about dimensionality of data, and specifically, since we work in biology, I will have some examples of biological data, like gene expression. And I want to talk about two algorithms that can be used to reduce the dimensionality of data to help visualize and analyze it. And I will talk about some of the problems that arise from high dimensionality that we encounter. So like one of these famous problems is this curse of dimensionality. And it depends on what field you come from, what people actually mean with it. So if you come from the machine learning world or like from statistics, you see the curse of dimensionality as this increase in the combinatorial space of your data, right? So let's say you have so you have some object that can take on 3 different properties in a different scale. That can already cover a pretty large space, and you would have to model this if you want to look at it as a statistical point of view. So this joint distribution is kind of like a 3-dimensional object, right? So if you add another dimension, you just exponentially grow that joint distribution space. The problem is you want to learn things from data because you can't really sample the whole data space. The large amount of dimensions, you will basically work with very sparse, view of the possible data space that you work with. And I have some examples for this later on. And then another thing is that things like the Euclidean distance do not really work in high dimensional spaces. And it's not like a really trivial thing to see. So there is example where let's say you have a cube here inside that cube, right? So it's basically, it has a radius of r, which is half the length of the edge of the cube. The problem is that the volume of a hypersphere, so this is for a 3-dimensional object, and have a 4-dimensional cube, a 5-dimensional cube. The volume of a hypersphere is like this formula here. We have the gamma function, which is just and then you have the volume of a hypercube. And actually, if you look at the ratios and you go with d towards infinity, the volume of the hypersphere gets it gets very small, right? So basically, the ratio between the two, the size of the dimensions. And what that kind of means is, like, if you look at that plot, is that all your data points will lie in the edges, will not be in the center, but they will be outside. If you have these data points in this Euclidean space, what this means is that the distances between these points are kind of all the same because they're all like pushed to the outside. So it's just it's a weird like geometric effect that you get from space. So in general, like we have to kind of handle dimensionality like in the most practical sense. It's really visualize 1,000 dimensions at the same time. We basically fail after 3, right? So 3 dimensions you can still play around with and see it, but then after that it's over. So what can you do, right? So there's a couple of things. This domain knowledge. So this is, like often used, let's say if you want to analyze images, so some expert will just come up with to take an image and extract certain like very precise features, and then you just work with those features. So you don't just work the pixels individually, right? The complicated feature space that you would have. So there's like things like SIFT, or like if you work with neurons, this is like shoal measurements that you can do, and you can kind of see how much neurons branch out. So you kind of try to map it down to some higher feature that is engineered, so you can come up with that concept. Another option is that you make assumptions about dimensions. So this is important for machine learning. The biggest assumptions you can do is that you assume independence of your variables. Because if you do that, then instead of looking at the big joint distribution, kind of look at the marginal distributions of the data that you have. So you can kind of get a much better image of, like if you sample data, you don't have to look at as much data because you don't have this exponential growth in your data point space. So if these are like naive Bayes or Bayesian networks, I think they were already mentioned in this lecture series. Other things, so you can kind of do smoothness where you, if you have some kind of discrete variables, you can kind of, if you have a data point in one bin, you might, want to smooth it out, so you like add additional values to the bins that surround it, so you can have some smoothing kernel. It's another method. Or if you have are, let's say, separated by time points, you can assume like a symmetry that you kind of ignore the order. But then what we mainly want to talk about today is that we want to reduce dimensionality of the data. So a couple of ways that can be done. So I specifically today talk about principal components analysis and t-SNE, which is a little bit different because it uses properties in your high-dimensional data space to kind of for this high-dimensional data in a low-dimensional space. And it has a couple of different ideas, but so like as a super simple example, this is like one of these classic physics problems. So imagine you have some spring and you have a monkey suspended from it. No friction in the system, right? So if, let's say, go and then the monkey will like kind of be attracted by gravity and then it will be pulled up again by the force of the spring, basically oscillate forever, right? Because there's no friction. So the monkey will just go up and down in dimension, right? So even though we're like in a three-dimensional space, we basically just have a movement that goes up and down, right? So system, and so this is kind of representative for anything that we want to measure, right? So this system could be, biological system, like how does a cell regulate itself? So this is just a really simplified way of some dynamic system. So let's assume we don't really know what is going on, right? We want to measure and understand what happens, right? So now we can have multiple angles of looking at this system, right? So let's assume we have 3 different cameras and they're just like placed randomly with different angles around the monkey, right? And each camera measures a dimensional image for each time frame. We have, so now we can collect data points. For every, let's say, second, we get 3 images, right? So, like now, how much dimensions do we measure per second? Any idea? Like our data point, like how many dimensions does it have? No. So we have 3 cameras, and each measures 2 dimensions. But when we have each camera measures like an X and the Y position of the monkey. And so, if we don't know anything about the system, we basically have a measurement for each point that has 6 dimensions, right? So we can already guess that we probably collect too much information because what we would expect is, because the monkey only moves in 1 dimension, really, this whole system only has 1 degree of freedom. But we measure it in 6 dimensions. We can expect this that, whatever we measure here, this must be somehow related to each other. So this is the thing. So there is something like what we call redundancy or correlation, let's say, between variables that we observe. So let's say we have 2 variables, X and Y. Yes, they're just independent, they're like randomly distributed, there's no, like if we know X, we don't really know anything about Y, right? So there's no mutual information between the 2 variables. something like a medium redundancy. I mean, it's not a, but there's some redundancy. If I know X, I know what Y is doing. Right? Extreme, where like if I know X, I pretty much know what Y looks like right away. Right? And so we have data, like a lot of data that we work with, especially gene expression, with the last type. So most of the dimensions that we have very redundant information. Okay. So what is the idea of, so this is also the idea of principal compound analysis. So we kind of want to find in our dimensions that we measured and a new space for our data points where the dimensions have no correlation. So there's no redundancy between the dimensions. And so this is, we can kind of take, this is kind of like an idea if you want to remove noise, for example. Let's imagine we have some matrix of, let's say, gene expression or whatever matrix we have, where we have samples as columns and rows. So this is matrix. So it is fairly complex. It's, some underlying low-rank matrix that carries most of the variance of the data. And then there is some sparse matrix that has basically noise. So this would be an idea. So like PCA could, you know, split these matrices apart and identify like random noise to some structured high-rank, low-rank matrix. So there's multiple ways to do this. So there's one that's like a singular value decomposition. So you can kind of break up every matrix into these. And what these do is basically a base transform base. And the nice thing is that you can truncate parts of it. So if you see in the middle, like where we had a 4x4 matrix, it's one of the rows and columns. And then if we calculate that back together, we get a very good approximation of the original matrix D with much compressed data. So now as a concept, let's say we have only like a tour example of 2 dimensions. X and Y. These could be genes, right? And their expression over like a set of samples, right? So each point is a sample. We can already see that, you know, we already have a lot of information by just looking at X because Y is so simple, right? What we might want to do is kind of, you know, principal component tries to kind of find a new dimension. So instead of X, space, find like the diagonal that kind of cuts through our data point, like in this example, right? So what this does is if I would project this down, so I can, you know, obviously I can take all these points and project them down to X, or marginalize them to X, or I could marginalize towards Y, right? But I kind of maximize the variance that's captured in the data, right? So my goal is kind of to find new dimensions that maximize the variance in our data. And so this is this red line, right? Red line, the data is spread out the most, right? And then the second one, we would just take an orthogonal line that goes away from this dimension. But it has much less variance than the first principal component, right? We have the more of these principal components we can define, right? So we kind of have to picture this like this data, like if we have like a high dimensional data set, it's kind of like an ellipse, like a high dimensional ellipse goes around the data space. This principal component values are basically, or these eigenvalues of the covariance matrix are basically the radii of this ellipse, right? Okay, so what we can do is like once we identified this, we can kind of treat the data points to these new dimensions, right? So basically, if you ignore the plot around it, this would be, you know, 2 new dimensions, which are the red lines, right? So this would be our new x dimension, and this would be our new y dimension. And then the data points get points in this dimensional space instead of the Okay. Later, just for the interest of time. So there's different ways to calculate these. Or if you use R or something, it's just a one-liner. So just to quickly say, like how you would usually do it is, you take your data points, which is a matrix, the covariance matrix. For your data points, eigenvalues, then the eigenvectors, and so the eigenvectors are your new dimensions, and these are the like scaling factors that tell you the size of the ellipse. And that's basically it. So it's a super simple mathematical approach, and this is why this is so popular. Well, I just want to like say like simple, so change the mathematics of this. But look how this looks like, right? So one of the key properties of this principal component is that, as I said, we want to find new dimensions that maximize the variance, the correlation between the dimensions. So there should be no redundancy between the different dimensions that we have. So the property is that we can then order these principal components by how much variance they capture. So the first principal component will capture the most variance in the data. The second component will capture the second highest, and so on. Let's say if I just create, so in blue you kind of see like code in R, just build like a random 1000 by 1000. And so then I calculate the principal components, which is this like PR comp. And now I plot the variance that's captured by each principal component from the highest component with the highest variance captured to the lowest. Random matrix, right? And what we can see is that the first principal component most amount of variance, and then we kind of get like a linear decrease. Like with every following principal component, it will decrease a little bit, right? On completely random data. So now let's say we build something quite as random. So let's explain this. Okay, so what I do is first I build a matrix that has, again, by 1000 random values. And now I build a new matrix where I 10 random columns from this matrix, and I take the average of them. And I use that as a value. So clearly, in our new matrix, some columns will have similar elements than others, right? The linear combination of this first matrix that we built. So there's some redundancy in there. So this is kind of plotted here in the red line. So you can kind of see that now the first principal component has actually a higher percentage of the total variance. The second one and third, and it's not a linear relationship anymore, but it's, you know, even more. And we can keep doing this, right? So now let's say here in the second one, so in my original matrix now only has 500,000, right? And now I sample from these 500 random vectors to build my new data points. The green line, so what we see here is now it's even steeper, right? So the first principal component captures a lot more variance than before. It's an even sharper drop-off. And like what's more interesting is if you actually look, green line, you can kind of see that after 500 principal components, like all the other components have 0 variance. So actually, even though we never told it that we built this data with 500 different vectors, so component analysis shows us that data is limited to 500 dimensions. After that, there's no principal components anymore that carry any. So, and then, I mean, just for the purpose of completeness, so now let's say I build the same, but I only start with 200 random vectors. So you can now see it's even sharper, and like after 200, there's no principal components. So this is pretty nice because it's just, so with this method, what's remarkable about it is that it's completely parameter free, right? So we don't do any assumptions. We just, the only thing we want is, we want to transform our data to a space where the dimensions are maximizing the variance and have no correlation. It's the only thing we demand, and like everything falls into place, right? And it's nice that we can, without doing anything, you know, it tells us exactly the dimensionality of our true underlying data. So where do you think gene expression falls in this picture? Any ideas? So if we would take like, I don't know, 2,000 samples from RNA-seq, from like random samples, what do you think this would like fall? Any ideas? Just a guess, you know? What?
I mean, I used this before with like gene expression data.
Yeah.
You just get an eigenvector for each gene, and then you can correlate it to your patient or some other quantity, but it gives you one value for across your entire data set for one gene.
Good point. So this method, so if you have a matrix, right, you can, it depends what you see as a dimension, you know. The genes are the dimensions, or you can say the samples are the dimensions, right? It really depends what the questions that you ask. The complexity of the data, right? So let's say with the blue, green, and red lines, you know, the more complex your data is, or more linear the distribution of the eigenvalues should be of your matrix, right? Like for gene expression, what would you expect? Like where would that line fall? Do you think it's like more complex than like 200 samples? So here I had only 200, right? Here 500, here like 1,000. If I just sample from completely random genetic backgrounds.
You take each gene as a feature, then would end up being 20K.
So that's what we observe, right? So we look at our original dimension is 20,000. But what do you think the real dimensionality is? Right?
It should be smaller because not all. Well, it depends on what you're asking, but not only for what you're looking for. What you're looking for is really data, maybe split with few bits.
The question, right? So the question would be like, ideally, like how many genes would you really need to capture all variants in all of gene expression? Like basically, it's the monkey experiment, right? So the monkey is super simple, right? But we have 3 cameras to look at it. So in the gene expression space, our cameras are the genes that we measure, right? But the dynamical system is much more constrained than the 20,000 genes that we look at, right? So the genes can't move independently from each other. They always move together. And so this is actually why this. So if you can see in the first 2 or 3 principal components, you explain 90%, 95% of the variability, right? If you want to understand the dynamics of cellular systems, the dimensionality is tiny. The vast majority of the variance that we observe in gene expression is, you know, explained with 3 variables. You know, you can probably separate by lineages of the cells. That's probably the biggest variance. And then after that, it's like, you know, very little, right? I mean, but there's still some percentage left. I guess there's no more. No. Oh, there is. Not at 0 here, right? But it definitely flattens off, and you don't gain much by measuring more genes.
Correct me if I'm wrong. When you increase the feature set size, doesn't that mean that every feature you can explain becomes less and less? So
Exactly.
Despite the fact that you have this huge spike at the beginning, they all contribute a really small amount.
Right. So the thing is that so a principal component in the end is a linear combination of genes. So it's like a what do I call it, right? So it's like a new dimension, which is just a mixture or combination of genes, right? And so yeah, in the first three combinations of these eigengenes explain a lot of your variance, and then after that it's just, you will find some eigengenes that still have a little bit of, information in it, but as you can see how it flats off is like it's tiny.
Right. What I was asking is like, you can explain a lot, but isn't that relative to like what I expect is like maybe 1 or 2% explanation if you do or
What do you mean?
so when you do PCA, like let's say the top 3 component, they would only
No.
explain a huge amount.
Yes, they explain like 95%. Basically everything you observe. 5% is where the small like differences come in. Where it's probably worth looking at 1 or 2 genes. Well, if you look at lineages or something, you have these markers, right? Like CD, I don't know, 75. It's only like whatever they're called. Yeah.
So what you're showing is that as you reduce the amount of data that you have, comprises of more of the variance.
Yes, exactly.
But in the bottom, you have 20,000, which is a lot. And it's because the data is correlated.
Yes, exactly.
So
the thing
is totally random.
So this is the thing. So this is how these, let's say for the blue one, right? So for the blue one, what I did is I start off with true random. I only have 200 true random vectors. 100, I combine them randomly to get, right? So I kind of expand the true variance is these 200, and I just redundantly reuse this variance to build more data.
So you're creating numbers that are correlated.
Exactly. So I could create 1,000, 10,000, 200,000. This complexity would never go up because I always sample from the 200. And so if I look at, so our 1,000 now is basically our genes, but they're really sampled from a much smaller set of true variants, right? And so this is why, like in the bottom picture, generated, variance distribution if you want. Right?
But it is a good idea to potentially, from the RNA-seq data, and then plot, like sample, take 1,000 genes and 1,000 samples, and then maybe get a line that fits on the first graph, just to see on the same scale.
I mean, this is nearly the same scale, right? This is just 2,000 samples overall genes, right? It is fairly comparable. Like it's not, if you only have 1,000 genes. I don't think it's. I think it's fairly comparable. So yeah, so what is like principal component used for? I think you see it a lot in applications, right? So one of the nice features is, you can't really show loads of, vectors that have 22,000 values, right? So when you take this matrix, principal components analysis, you can only, let's say, plot the first 2 principal components. So the first 2 principal components, by definition, carry most of the variance, right? So in the example, if I go back, the first 2, I capture, some significant percentage of the total variance. So it gives me like a good idea of what my data distribution looks like, right? And I can kind of project additional information on it. So here, paper where you can kind of see, systems versus other tissues, and then they also have a separation between normal disease, neoplasm, and cell lines. Right? So it's just easy to break that apart. So so this is the idea, right? So you start with a big matrix where you have, I don't know, let's say 5,000 samples and 22,000 genes. Just this big thing, you can't really look at it by eye. You can kind of have a nice view of it. Okay. Yes.
You're graphing that. Your axes. Are you determining like the content of the axes? Aren't you just doing PC1 versus PC?
Yes, exactly. So in this one, they just plot the first and second principal.
And then they look at the data and determine that that's how
they annotated them manually, right? Obviously, the algorithm wouldn't explain you what the difference is,
right.
But they just looked at it and then they determined.
But they used the entire data set. It's not like they subset the data set.
No, yeah, it's the whole data set, right? What you really do is you still have basically the same number of samples, right? But the samples live in a different dimensional space, right? And then you just plot the first two dimensions, which carry most of them. Yeah. It's kind of like, I mean, you could also, I mean, technically you can also just take the samples and take two random rows from your gene expression. Genes, right? It's very commonly done if you look at marker genes, right? Or like if they do some kind of selection on, basically have these scatter plots just showing the expression of two genes.
Flow cytometry.
Right. So you can, you can just use two genes as a form of feature selection. But you can, this is, I guess, more advanced because you features and maximize the variance. Okay. And then, so this was an interesting observation that Neil Clark did, that was working with Avi like last year. I think he left a year ago, pretty much. He looked at breast cancer samples. And so you have these different subtypes of luminal and luminal B, basal, and so on. And so he kind of shows that there is a difference in their, which is kind of interesting. So you can kind of see that if he plots the principal component dimensionality, you kind of see that which one has the lowest complexity, like normal-like have the lowest complexity, and then basal-like have the highest complexity. And it's kind of preserved through the three different ways of measuring look like. Right? And it's also interesting to see if you compare, like, let's say, transcriptome, like expression. That expression actually has a much higher dimensionality relation. But this, I think this you could also capture by just asking how much variance you observe. I think that's a similar measure. So it's just an example how you can kind of look at complexity of data and how it differs between different types. So now I have like a completely different example. So this is like a data set. It's like the MNIST data set. And basically what it is, it's just a large collection of handwritten digits. Popular data set for machine learning because it's kind of manageable, right? So it only has 28 by 28 pixels. And then you have grayscales. Of those. So, and by now, so like then you have these challenges where they write neural networks and they try to predict the correct digit. And so they get to like 96% accuracy, which is better than humans, I think. I mean, they're getting pretty good. So, one of these cases where is important. So this problem and its dimensionality, so this is 28 by 28 pixels. have 784 dimensions, digit, right? And then you have these gray levels in there. So like even if you would only have black and white, the sample space, or like the true space, is 2 to the power of 784. Obviously huge, and because if you have all these, I mean, these are random images in this space, right? Do with these numbers that you want to learn. Even care like to look at something like this, or assume that what you look at is something like this, because it's completely different. Right? So now, I mean, we talked about principal components. So now we could ask, well, can principal components take this 784 dimensional space, you know, map it into 2 dimensions, and then we get like nice clusters between the numbers, right? So it doesn't work, right? So you kind of see the points are basically colored by the digits that they represent, and the principal component analysis doesn't really, they can't really figure it out. It's because you know, it will try to find some like similarity between these fields, but the thing is, like, you know, a 0 could be slightly shifted over, might be shifted over, so there's no real overlap in the pixels. So, and because the principal component analysis doesn't really have an understanding, you know, of the shape or the form of that, this method doesn't work. So in this case, principal component analysis is not a good method.
Separating the data.
Yeah. So so it's a slightly different problem, and it's basically so one of the other challenges is if that you work with has some local structure to it, then principal component analysis might not be the ideal way to do it. And so this picture kind of illustrates this quite nicely. So on the left, you have this manifold, it's called. So it's some high-dimensional space. So as you can see, this like whatever like snail-like pattern, local structure to it. So you know, the points in red, they're kind of close together. The points in orange are close together. But so you can also see that there's orange and blue is right next to each other. But like as a local property, they should actually be far away. So if we would try to project this to a 2-dimensional space, what we really would like is something that we see on the right. So we don't want to see orange next to blue or like next to red. We want them to be apart. So we want basically the method to understand this kind of shape. And so this is a different algorithm. It's called like t-SNE. And it's basically trying to calculate an embedding of high-dimensional data to low-dimensional data by preserving the original distances between objects in the high-dimensional space. So the distances should be the same. So how does it do that, right? So it's should actually do it a little bit different in the implementation, but simply speaking, what they do is here. So this algorithm actually has a lot of parameters, like that, like describe how big the neighborhood of a point is. Let's say I'm here, like in a blue point. So what it will do is it will calculate in the distance around this blue point neighbors and weigh them, or like, and then so it calculates the distance to, let's say, one neighbor, and then normalize it by the sum of its vicinity. So you kind of weigh other, and by doing that, you kind of ignore, so like points that are far away or not in the direct neighborhood in that distance, and only things that are close will have like a significant like closeness to it. Kind of allows you to preserve this like local structure, and by this definition, they can basically use this transient behavior of the local similarity. So because you can go to the next, and this point has again like new neighbors, and this point has new neighbors. So you kind of carry the structure of the high dimensional space into the low dimensional space.
Do you determine the space
Um,
to do that? But you have because
so there is a bunch of things. So they use some kind of force. I don't know if you know these networks when they kind of self-arrange. Did you ever see that? And they other way or like pull at each other. And so they have parameters like that. So it's kind of like it's an iterative process, right? So you first, you have the instances before, and then you have some random assortment at the beginning of your data points. And then you calculate the stresses, like let's say one point is very far away from its neighbors in the high dimensional space. Then in the low dimensional space, these points will attract it, kind of like self-assorting the low dimensional space, given the high dimensional space. That's kind of the idea. And then they basically try to minimize the KL divergence between these two values. So terms are literally just the distance of its neighborhood in the high dimensional space to the distance of its neighbors in the high dimensional space and the low dimensional space. It's kind of, and this is kind of like if you visualize it, like what it does. There's some kind of phase transition. I don't know why it like expands at some point. This has something to do with implementation in R. Because at the beginning it's like very tight and then it kind of blows up. But so this is for the same data set that I showed before, right? So where principal component analysis wasn't able to find any nice structures or clusters, this algorithm is actually pretty good about it. Like, I mean, there's some mistakes with the 9, like this 9 cluster that kind of gets pushed out, but overall it looks much better than before, right? This is because handwritten letters do have this, they do have this local structure. And I can maybe show it in a picture here. So what you can see, like in the bottom right, you can kind of see that the digits are perfectly separated. So here they probably use better parameters than what I chose. And they had more data points. They have the much finer, like, your understanding of the local structure of your data is. So I think this is why this algorithm performed better on this data set than mine. But see, I don't know if it's, let me see if we can. I think is this where are the ones? I think these are the ones that they're kind of like here, the ones are all kind of tilted like this. And then they slowly, tilt like towards this side. Kind of starts here and like it knows, so this is a neighbor of these, and then this is a neighbor of these, so they kind of all hang together in this local structure. And this is true also for like, these letters, they tend to be kind of tilted to the left and then they're tilted to the right. So this is an example why this like local structure in your data can really help you define neighborhoods that you would lose in something like a principal component analysis, because there every point, is independent of its like similar data points. So it's not what you would, it's a completely different idea.
Is drawn on the PCA part to show you some initial structure. Within the drawing of, let's say, a 1, drawn on this angle there on the top, and then the ones drawn that way there on the bottom. So this is just a pretty idea of like combining PCA plus original images.
Yeah, I think this. I mean, it's a nice idea because you can kind of see what the algorithm is actually doing, right? So it kind of you can really see, oh yeah, it actually. So, I mean, this is the thing, right? Like even though at this point, like let's say this one and this one have nothing in common, because they have some connectivity somehow.
So, what is the because you've seen this type of dimensionality expression data as well.
So for expression data, so this is the question, right? It really depends. Data people mostly use PCA.
Right.
And there's no reason necessarily why t-SNE would be worse. I feel like, I mean, you often hear, oh, biological systems aren't linear, you know, like what, like it's a big constraint to use PCA. But to be honest, gene expression is pretty linear. Between genes are largely linear. PCA works really well. And the question would be, so let's say your data points would show this kind of local neighborhood, like if they describe some like geometric structure in this high-dimensional data, would do better in reducing it. The problem is that in t-SNE, what you get from applying this method is some arbitrary values. So these values don't really. So let's say like this, like just because this point is here and this is here, this doesn't tell you anything, right? You can't say that this. I don't think you can really say that this is closer to this than this to this or something, right? It's like interpret this is a bit different. Like in PCA, because you just do a linear transformation and you don't have any assumptions. I think it's a safer transformation of your data, creating much nicer pictures to look at. But you know, yeah, if you want to just visualize your gene expression, it's fine to try both. And I've seen t-SNE working much better on gene expression than PCA did, for sure.
I always see PCA on gene expression, like RNA-seq data, but if you do single cell, like always t-SNE.
I think this is one of the reasons, like in single cell, I mean, so people say single cell has this firstly gene expression, right? Where if you have translation of a gene, you get like a lot of mRNA for like a short window of time, and then it disappears, right? And so over like, let's say your 22,000 genes, if you want to compare individual samples, right? But you have this local structure, so if you have a lot of single cell samples, they're just different because of noise and whatnot. Wholly identify commonalities of the global structure and then use that to build your cluster. Right? Yeah, I don't know. I mean, it's hard to quantify what's better or not, right? Like I would try both. If you just want to visualize it, you just should run both and see which one looks better. Honestly, but I mean, just conceptually, I hope it's clear that like, so this is kind of using this. PCA is mostly preserving these large differences between your data.